Welcome to Cell Biology. Today, we are exploring two of the most fascinating organelles in eukaryotic cells, lysosomes and peroxisomes. Think of them as the microscopic waste management and hazardous materials teams of your cells. Without them, our cells would quickly choke on their own debris or be destroyed by toxic byproducts. Let us dive in and see how they keep us alive. Now that we know what we are looking at, let us start with the lysosome. Imagine a bustling city producing tons of waste every single day. If there were no garbage trucks, the city would cease to function. The lysosome is the cell's essential recycling center. It takes in damaged cellular parts, invading bacteria, and large food particles, breaking them down into reusable building blocks. Without this tiny but mighty organelle, the cell would literally choke on its own debris and die. It is a critical system for cellular survival and efficiency. To understand how this recycling works, we have to look inside. The lysosome contains a highly concentrated mixture of over 50 different degradative enzymes, known as acid hydrolases. These include proteases, nucleases, and lipases. But here is the brilliant part. These enzymes only work in a highly acidic environment. This acts as a built-in safety mechanism. If a lysosome accidentally breaks open, the neutral environment of the cytoplasm inactivates the enzymes, preventing them from accidentally digesting the cell itself. This ensures the cell remains completely safe from its own destructive tools. This brings us to why lysosomes are so critical for human health. They perform a process called autophagy, where the cell digests its own obsolete organelles to survive periods of starvation. In our immune system, specialized white blood cells use them to destroy dangerous bacteria. But when these enzymes are genetically defective, it leads to severe conditions called lysosomal storage diseases. A tragic example is Tay-Sachs disease, where missing a single lipid-digesting enzyme causes a fatal accumulation of fats in the brain. Knowing how important this acidic environment is, let us look at the mathematics behind it. The lysosome uses an ATP-driven pump to move hydrogen ions inside. The concentration of these protons determines the pH. In this formula, pH represents the power of hydrogen. The symbol H+ in brackets is the molar concentration of hydrogen ions. Because of the negative base 10 logarithm, as the concentration of hydrogen ions increases, the pH value actually decreases. So a lower pH means a more acidic environment. If the proton concentration drops, the pH will rise, making it less acidic. Let us see this formula in action with a concrete example. We are given that the cytoplasm has a neutral pH of 7.0, while the lysosome interior has an acidic pH of 5.0. We need to find how many times more concentrated the protons are inside the lysosome. First, we rearrange our formula to solve for hydrogen ion concentration. Substituting the values, the cytoplasm has 10 to the negative 7 molar protons, and the lysosome has 10 to the negative 5 molar. Dividing the lysosomal concentration by the cytoplasmic one, we subtract the exponents. The result is 100. This means the lysosome actively pumps protons until it is exactly 100 times more acidic than the rest of the cell. Now that we have calculated the gradient, let us visualize how the cell achieves this. Here you are looking at a schematic of the lysosome. Notice the single phospholipid bilayer membrane separating the neutral cytosol on the outside from the acidic interior. The key feature is the ATP-driven proton pump embedded in the membrane. It is actively forcing hydrogen ions, shown as H+, into the lysosome against their concentration gradient. Inside, you can see the acid hydrolases, now fully active in this pH 5.0 environment, ready to break down cellular waste. Having explored the general recycling center, we now move to the cell's specialized hazardous waste treatment plant, the peroxisome. As your cells burn fuel for energy, they accidentally produce highly toxic chemical byproducts. One of the most dangerous is hydrogen peroxide, which can shred DNA and proteins. The peroxisome acts as a secure, isolated chamber where these dangerous chemicals are safely contained and neutralized. Without this vital protection, the cell would quickly destroy itself from the inside out. To understand how peroxisomes neutralize these threats, we must look at their structure. Unlike lysosomes, peroxisomes have a dense crystalline core packed with oxidative enzymes, most notably catalase and urate oxidase. These enzymes strip hydrogen atoms from organic substrates and transfer them to oxygen, which ironically produces toxic hydrogen peroxide as an intermediate step. But because hydrogen peroxide is so dangerous, the peroxisome immediately uses the enzyme catalase to convert it safely into harmless water and oxygen gas. Beyond just handling hydrogen peroxide, peroxisomes are absolutely vital for overall metabolic health. They are heavily involved in lipid metabolism, specifically breaking down massive, very long-chain fatty acids through a process called beta oxidation. In your liver, they play a crucial role in detoxifying alcohol and other harmful compounds from your bloodstream. When genetic mutations prevent peroxisomes from forming properly, it results in devastating conditions like Zellweger syndrome, where toxic fatty acids accumulate and destroy the brain and liver. Now that we understand the vital role of these enzymes, let us look at the formula that quantifies their protective power. Vmax represents the maximum velocity, or the maximum rate of the reaction in molecules converted per second. The symbol kcat is the turnover number, which is the maximum number of substrate molecules a single enzyme can process per second. Finally, ET in brackets is the total number of enzyme molecules present. This shows that the total processing power increases proportionally if either the speed of the enzyme or the amount of enzyme increases. Let us apply this formula to see just how fast a peroxisome works. We are given that catalase has a staggering turnover number of 4 × 10⁷ molecules per second. We are also told a single peroxisome contains exactly 500 catalase molecules. We need to find the maximum number of hydrogen peroxide molecules it can neutralize in 1 second. By multiplying the speed of one enzyme by the total workforce of 500 enzymes, we find the total maximum velocity. The result is 20 billion molecules per second. This staggering number proves the cell is perfectly equipped to handle its own toxic byproducts. Now that we have calculated its incredible speed, let us visualize this detoxification process. Here is a schematic of a peroxisome. You can clearly see the outer membrane enclosing the dense crystalline core where the enzymes reside. Notice the specific reaction taking place. Toxic hydrogen peroxide, labeled as H2O2, enters the enzyme catalase. Instantly, it is broken down and released as harmless water and oxygen gas. This simple but incredibly fast reaction is what keeps our cells safe from oxidative damage every single second of our lives. We have covered a lot of ground today, so let us summarize the most important ideas. First, the lysosome is the cell's indispensable digestive system, using a highly acidic environment and proton pumps to safely break down complex molecules. Second, the peroxisome is the hazardous waste plant, utilizing incredibly fast enzymes like catalase to instantly neutralize toxic byproducts like hydrogen peroxide. Together, their unique abilities to compartmentalize dangerous reactions make them the unsung heroes of the cellular world, indispensable for maintaining cellular integrity and human health. Great job following along.